Hi, everyone. Thank you for joining us today for the sixth webinar in our EPIC series on the use of NAMs in risk assessment. This series is being co-organized by the Environmental Protection Agency, PETA Science Consortium International, the Institute for In Vitro Sciences, and the California Department of Pesticide Regulation. I am Amy Klippinger, president of PETA Science Consortium International, and I'll be co-moderating today's webinar along with several others, including Naladri Bhowmick, a toxicologist in the Human Health Assessment Branch of the California DPR, Rachel Bruner from the US EPA, and Christy Sullivan, the Vice President of Education and Outreach at the Institute for In Vitro Sciences. Please note that the slides and a recording from today's webinar will be posted online at this link shortly after the webinar. All future EPIC webinar materials will also be available at this link as the series continues. We also wanted to ask you to save the date for our next EPIC webinar, which will be January 29th, 2025, hard to believe. And it will feature Dr. Katie Paul Freeman from the U.S. Environmental Protection Agency Office of Research and Development and Dr. Elizabeth Berggren from the European Union Joint Research Center. They will be talking about assessing systemic toxicity with NAM-based approaches. You can register now for that webinar on the Science Consortium website at the link that was shown on the last screen. So we have two speakers today who will be presenting on regulatory tools for assessing the skin sensitization potential of chemicals and a case study using the Sarah Ice model. First, we'll hear from Dr. Donna Macmillan, Director of Education and Regulatory Engagement at the International Collaboration on Cosmetic Safety. And second, from Dr. Emily Reinke, a Principal Predictive Toxicologist at Innovative and contractor supporting the U.S. National Toxicology Program Interagency Center for the Evaluation of Alternative Toxicological Methods, otherwise known as NICEDOM. We will do a Q&A session at the end of the second presentation. The webinar is scheduled for 1.5 hours, and we may wrap up earlier depending on how long the questions go. Everyone is muted, but you can type the questions into the question module within your Zoom toolbar at the bottom of your screen. And please do indicate if your question is directed to a specific speaker. And also, we won't be monitoring the chat, so please don't put your questions in the chat. Make sure they go into that question module. We've also enabled closed captions. You can turn this feature on and off according to your preference using the toolbar at the bottom of your screen. Before we start the presentations, our colleague from the California DPR, Naladri Bhowmick, will share a few comments. Naladri?
Thank you, Amy. Hello, everyone. So on behalf of here at DPR, I extend a cordial welcome to all of you to the sixth edition of the EPIC webinar on NAMs. I must mention here that California DPR are excited to be part of this webinar series from the beginning, along with US EPA, IIVS, and PETA. On behalf of the panelists here, I extend my warm welcome to our speakers today. And since the announcement was made, we are all eagerly waiting for these two presentations on developments and approaches to implementation of NAMs on dermal sensitization. No doubt an important topic to regulatory agencies, including our work at Human Health Assessment Branch at CDPR. With those words, I return it back to Amy.
Great, thank you, Naladri. And I will now turn it over to Rachel Bruner, who will present a few opening slides on the US EPA perspective on skin sensitization testing. Rachel?
All right, thank you, Amy and Naladri, and good morning, and thank you to everyone for joining us today to discuss skin sensitization. To set the stage, I would like to briefly share EPA's perspective on skin sensitization testing, specifically related to regulatory submissions in EPA's Office of Chemical Safety and Pollution Prevention, or OCSPP, where our mission is to protect human health and the environment from potential risks due to pesticides and commercial or industrial chemicals. We can go to the next slide, please. Our disclaimer, the views are those of myself and do not necessarily reflect views or policies of the US EPA. Next slide. Okay, so in 2018, EPA OCSPP released the interim science policy on the use of alternative approaches for skin sensitization as a replacement for laboratory animal testing. This document was developed in collaboration with the Interagency Coordinating Committee on the Validation of Alternative Methods, or ICVAM, and the NTP Interagency Center for the Evaluation of Alternative Toxicological Methods, or NICETM. And it builds on international activities with member countries of the International Cooperation on Alternative Test Methods. The science policy applies to both submissions to OCSPP's Office of Pesticide Programs, which implements the Federal Insecticide, Fungicide, and Rodenticide Act, or FIFRA, as well as OCSPP's Office of Pollution Prevention and Toxics, where I work, OPPT, which implements the Toxic Substances Control Act, which covers industrial chemicals. OPPT does not have I should go back. So OPP for pesticides, skin sensitization testing is generally required. Whereas for OPPT, we don't have any specific data requirements per se, but we do receive skin sensitization information, mostly for chemicals in the new chemicals program. However, if a chemical is suspected of being a skin sensitizer, we can request that information of the submitter. Both OPP and OPPT are committed to developing and implementing new approach methods, and here specifically encouraging the submission of non-animal studies for the skin sensitization endpoint in lieu of the LLNA animal test. And, although this policy document is a draft for public comment, given the substantial science evidence and international activities supporting NAMs for skin sensitization, OPP and OPPT have been and continue to accept these approaches under the conditions described in this document. Also in this document, we can go to the next slide, please. The adverse outcome pathway for skin sensitization is covered, and we're going to talk about this several times over the course of this webinar. I just wanted to briefly introduce it. It describes the key events that occur during skin sensitization, from, the molecular initiating event to cellular response to organ and organism level response. And then overlaid on this diagram are the OECD test guidelines that cover testing for the different key events in this process, with non-animal approaches for key events 1 through 3. And non-animal testing results from these key events can be integrated in what is referred to as a defined approach to make a prediction for skin sensitization. And I look forward to our speakers diving into this in more detail. Next slide, please. We'll be talking, a lot about defined approaches today. So just a quick definition. A defined approach is a rule-based data interpretation procedure applied to data generated with a defined set of information sources. In this case, results from assays designed to test for the key events, presented in the previous slide, to derive a prediction. And in case you're interested, an evaluation framework for skin sensitization defined approaches is also provided in this policy document. And, no webinar on a topic on NAMs would be complete without discussing, the variability observed in animal studies. Here, the reproducibility of the LLNA is in the range of 70 to 80% for hazard prediction and 60 to 70% for potency prediction. I say that just to point out there's some inherent uncertainty when comparing the defined approach predictions to the LLNA endpoint. But this is, still a common baseline for predictive performance of defined approaches. That said, of the existing 6 defined approaches evaluated at the time of the development of this document, which is 2018, all were comparable or better in terms of performance to the LLNA. Okay, so thank you. The 2 defined approaches included in the policy document are on the left, the AOP 2 out of 3, and then on the right, the key event 3.1 sequential testing strategy. And these will get discussed more today. So with that, I would like to thank our 2 speakers for all of their efforts and continuing to push the work in the skin sensitization space forward, and also thank my co-organizers for all the work they do to make these webinars happen. Okay, so with that, I would like to introduce our first speaker. Speaking first today will be Dr. Donna Macmillan. Dr. Donna Macmillan studied biomolecular and medicinal chemistry at the University of Strathclyde in Glasgow, where she remained to complete a PhD in organic medicinal chemistry, focused on the development of novel drugs for schizophrenia. She spent several years working in drug development. And then in 2013, Donna joined LASA Limited, where she specialized in knowledge-based expert systems and QSARs across several endpoints, including mutagenicity and skin sensitization. She then joined the Humane Society International in 2021, and Donna worked to advance a paradigm shift from traditional toxicology to non-animal approaches for chemical safety assessment. In 2023, Donna assumed the role of Director of Education and Regulatory Engagement at the ICCS. And in this capacity, she leads efforts within the education and training and regulatory engagement pillars to align with and leverage scientific advancements in cosmetic and chemical safety. Please go ahead, Donna. And your screen looks good.
Oh, great. Thank you for letting me know. And thank you so much, Rachel, for the kind introduction. And thank you to the organisers for inviting me to speak today. So I've put a brief overview of what I'll be talking about today on this slide. I'll begin by giving a little bit of context by describing what the OECD is. Then I'll talk about skin sensitization in general, the move towards non-animal approaches, including the development of defined approaches, and then talk a little bit more about some newer emerging activities that are under evaluation at the OECD level. So looking at the OECD, so the OECD is the Organization for Economic Cooperation and Development, and it was established in Paris in 1948. It consists of nearly 40 member countries, each who nominate an overall ambassador known as a national coordinator who represents the interests of their own member country. They also nominate country experts to serve on various working parties and committees. And one of the big or the main tasks of OECD is to develop and revise test guidelines. And these are documents that are used for testing of chemicals for human health and the environment. And it's a way of standardizing the way that these tests are carried out. And these test guidelines actually promote the mutual acceptance of data. That's sometimes known as MAD. And this concept allows the avoidance of duplication of testing. It reduces the number of animals used if a test guideline is for an in vivo test. And a phrase that's often used with respect to MAD is tested once, accepted for assessment everywhere. And what that means is that if a member country runs a test according to an OECD test guideline, then they can use that in another member country for their regulatory assessment. There are many different toxicological endpoints that are covered by OECD test guidelines. And of course, today, what we will be talking about is skin sensitization. But let's just take a step back and think about skin sensitization from a biological perspective. Now, of course, you're not here to hear the ins and outs of the biology, so I'll just go over this quite quickly. But it's good to make sure that we're all on the same page with respect to what we know about this endpoint. So skin sensitization is often known as allergic contact dermatitis, ACD, and of course it's often evaluated for as an endpoint for various regulations, either for hazard assessment and of course risk assessment of chemicals. And there are two phases to skin sensitization. The first one is the induction phase. And this is when your chemical, which is known as a hapten, it penetrates through the skin, and then it forms a conjugate with carrier proteins. And this conjugate, this hapten protein complex, interacts with keratinocytes and dendritic cells, which leads to activation of T cells in the lymph nodes. But the second phase is when skin sensitization actually occurs, and this is the elicitation phase. And after subsequent contact with the same chemical, this hapten protein complex or conjugate is formed again, and after a similar process to the induction phase, an inflammatory response occurs, and then it causes the adverse outcome of skin sensitization. Now, the adverse outcome of skin sensitization has been really well studied over the past couple of decades. And in 2012, the OECD actually published the adverse outcome pathway for skin sensitization as initiated by covalent bonding to proteins. And this adverse outcome is shown here on this slide on the left-hand side. And an adverse outcome pathway is a way of showing a series of biological events that leads to an adverse outcome. Just let me see if I can grab a pointer. Yeah, so it's a series of biological events that goes through certain key events to then lead to an adverse outcome. In this case, that's the organism response of skin sensitization. And this AOP, as I said, was published back in 2012 and was the basis of the development of many other in vitro tests. But when we look at traditional approaches to skin sensitization, there are two main OECD test guidelines that are used, and they're shown here on this slide. So on the left-hand side, we have test guideline 406, which is the guinea pig maximization test. And then on the right-hand side, we have OECD test guideline 429, which is the murine local lymph node assay, the LNA. And these cover key event 4 and the adverse outcome of the skin sensitization AOP respectively. So I've just popped here in purple the LNA and then the guinea pig maximization test under each of these. But when we look towards the non-animal approaches that have been developed over the past couple of decades, these actually cover different key events in the adverse outcome pathway. So if we look at this one here on the left-hand side, OECD test guideline 442C, this contains several assays that actually address key event 1, so the first key event in the AOP. And this test guideline currently contains the DPRA assay, so the direct peptide reactivity assay, the amino acid derivative reactivity assay, the ADRA, and the kinetic direct peptide reactivity assay, the KDPRA. We also have an OECD test guideline which covers key event 2, so that is OECD test guideline 442D, and that currently contains the keratosenescence assay, the Lusense, and Episense A. And then the final OECD test guideline that contains non-animal methods is OECD test guideline 442E. And this covers key event 3 and currently contains HCLAT, USense, the ILA luciferase assay, and guard skin. These can also be mapped onto the same adverse outcome pathway. And these are shown here in their relative positions. So OECD test guideline 442C under key event 1, 442D under key event 2, 442E under key event 3, and then, as we saw before, the animal methods on key event 4 and the adverse outcome. I've also put the in silico tools here. Often these are considered as measuring the electrophilicity of a chemical. So it's good to see that on the adverse outcome pathway. So you can see how all these different assays can be used in combination. And speaking of using them in combination, that's the purpose of the defined approaches for skin sensitization. So with the publication of these non-animal assays in 442C, 442D, and 442E test guidelines, these were used to develop some defined approaches. And of course, some of these have already been mentioned by Rachel. So these were originally published in peer-reviewed scientific literature and then published by the OECD as examples of how to report a defined approach for skin sensitization and how to report it within the context of an IATA, so an integrated approach to testing and assessment. And I'll just go over the definition again, although I know that that Rachel mentioned it at the beginning of today's webinar. But a defined approach consists of a fixed data interpretation procedure, sometimes known as a DIP. And this can be a mathematical model, it can be a rule-based approach, an algorithm that's applied to data that is generated with a defined set of information sources, where your information sources can come from an in silico tool, an in chemical assay, or in vitro data. And then this allows you to derive a prediction that doesn't need any expert judgment. So you will get the same prediction irrespective of who carries it out, as long as you're using the same data inputs. So this, of course, removes subjectivity and allows predictions from these defined approaches to be used under MAD. So if we now think about the original development of OECD guideline number 497, which is the defined approaches to for skin sensitization. Back in 2017, a project was submitted to the OECD and it was co-led by the European Commission, the United States, and Canada. And they proposed developing a guideline on these peer-reviewed, these defined approaches for skin sensitization that had been published in the peer-reviewed literature. So this was then added to the OECD's work plan, and the work began in 2017. So if we skip forward several years, consisting of much hard work by the expert group and all the member countries involved, In 2021, in June, the guideline was published. So this is guideline number 497. And this was really quite groundbreaking because it formalised the combination of several non-animal methods, so in chemical and in vitro tests, as well as the use of in silico predictions. And each of the DAs that are covered in this guideline perform better or equivalent to the LNA, which was used as the benchmark data. And then the two DAs that were included in this guideline were the 2 out of 3 defined approach and the integrated testing strategy, version 1 and version 2, depending on which in silico tool was being used. So I'll try and give you a bit of an overview of how the 2 out of 3 defined approach works or how you conduct it. Now this DA provides a hazard prediction, so sensitizer or non-sensitizer, and you use concordant results from up to 3 different assays from the DPRA, the keratinescence and HCLAT. And if you remember from a few slides ago, they cover key event 1, key event 2 and key event 3 of the skin sensitization AOP respectively. So on the right-hand side of the slide, you can see the figure that's published in OECD test guideline 497. And if you begin here, you conduct any 2 of the assays that cover the 3 key events. And if 2 are concordant and none are borderline, so say you have positive and positive, then you conclude it to be positive and a sensitizer. Similarly, if you have 2 negative results, then you have confidence in these and you conclude it as a non-sensitizer. It becomes a little bit more complicated when you have discordant results from your first 2 assays. So maybe one is positive and one is negative. And then in that case, you conduct the 3rd assay. And depending on the outcome of that, you apply the 2 out of 3 approach to come to your outcome of sensitizer or non-sensitizer. And if any of the results are borderline, which you can see the thresholds are stated in the guideline, then you may need more information before you can make a prediction. But if we look at the performance of the 2 out of 3 against the reference LNA and the reference human data, so if we look here on the left-hand side, this is the 2 out of 3 when we compare against the benchmark animal data, so the LNA data. And this is a confusion matrix that indicates how many are inconclusive and how many have been correctly predicted. This table here provides the performance metrics of accuracy, sensitivity, specificity, and balanced accuracy. But we'll just look at balanced accuracy for now. So balanced accuracy here is 84%. And then when we do the same performance calculations, but instead use the human data that we have, we see that the balanced accuracy is 88%. So really great performance here. And what's interesting to note is that when we compare the animal data against the same human data, we see a balanced accuracy of 58%. So as Rachel mentioned in her slides about the variability of animal data, and the fact that the defined approaches have been shown to predict actually equivalent or better than the benchmark animal data. Now moving on to the ITS, so the integrated testing strategy. Now, this is a little bit more complicated because there are scores applied to the different information sources. And there's also two versions, depending on which in silico information source you use. So if you are using Direct Nexus, then it's ITS version 1. If you're using OECD Toolbox, it's ITS version 2. And then you apply a score to the results that you get from the HCLAT or the results you get from the DPRA, and then the outcome that you get from your in silico tool. And then when you sum these scores, then using this table, that allows you to assign a potency prediction according to GHS, so 1A, 1B, or not classified. And I'll just go over how you apply that using the workflow. And this workflow is published in Guideline 497. And I've tried to make it more simple because it looks a little bit complicated when you first look at it. I've tried to help by numbering these boxes and then providing an explanation of how you assign it. But we'll see how we go on. So if we have if we're using the, if all of our information sources are in domain, or there's no limitations associated with the assay, then we use the scoring in box number 1. So in this case, we sum the scores from the DPRA, the HCLAT, and your in silico tool, and you assign the scores as I showed in the table on the previous slide. But what we're able to do with the ITS is actually use partial information sources. So where one information source is maybe out of domain, like maybe your in silico prediction is out of domain, maybe there's a limitation associated with your in vitro test that your chemical falls under. But in most instances, you can still actually derive a prediction using the ITS. And this is only when the overall, or sort of the missing information source score wouldn't have an effect on the overall prediction. So if we look at the middle box, number 2, so this is where the in silico prediction is out of domain. And in this case, all you have is scores from your DPRA and your HCLAT. And then in that case, you would use this table. And it shows that occasionally you will have an inconclusive prediction. And then similarly for the right-hand box, so box number 3, this is when one of the in chemical or in vitro assays is out of domain. And then in this case, you only use the scores from your applicable assay. So whether, depending on which one's out of domain, of course, it's the other one. And then your in silico tool. And then here you would use this table. And here there may be instances where you have an inconclusive result. And in that case, you may want to try and apply a weight of evidence within the context of an IATA. And there's some publications that try and describe how you can do that. But looking at the performance of the ITS, if we begin with the hazard, and just to be clear, this table here is ITS version 1, and this table here is ITS version 2. But when we look at the overall predictions and the balanced accuracy, we can see that for ITS version 1, it's 81% balanced accuracy, and for ITS version 2, it's 80%. So they both predict very well and almost the exact same. When we look at this against human data, so here we were comparing against the in vivo benchmark data. When we look at the human data, here we see that the balanced accuracy is actually 69% for both of the ITSs, so ITS version 1 and ITS version 2. And then again, just for comparison, when we look at how the LNA data predicts the human data. So this is for hazard. Again, we see that the balanced accuracy is relatively low in comparison to how well the defined approaches predict the same human data. But that was hazard. So now if we look at potency, so this slide is quite busy as well. So the left-hand side shows the results for ITS version 1. So that's the one using Derek Nexus, with LNA at the top and human at the bottom. And then on the right-hand side, we have ITS version 2, which uses OECD Toolbox as its in silico source. And again, LNA at the top, human at the bottom. But what we can see is that for ITS version 1 and ITS version 2, they both predict the LNA outcome with 71% accuracy, so correct classification overall. And then similarly, when we look at the human data, it's 68% for ITS version 1 and 70% for ITS version 2. And again, if we look at the comparison of how well does the LNA predict the human data in terms of potency, so the GHS categories, then that's a little bit lower than the defined approaches with 60%. So this slide just shows an overview of the results that were in the previous slides. You know, the other slides were quite busy, it can be quite hard to pull out the data. So I thought it might be nice to see this in a little table where you have the DA on the left-hand side, and then the hazard as when using LNA as comparison versus human, and a potency here. So that's just a nice way to look at all the numbers together. And now just moving on to emerging methods. So these are newer methods that are under evaluation at the OECD, or new projects that are currently under evaluation at the OECD. So, and these are designed to really increase the utility of the defined approaches or improve the applicability domain. And one such project is shown here on this slide, and this is the use of alternate OECD test guideline information sources. So this project was put together because currently the defined approaches that I've just described only use DPRA, creatinine, and HCLAT. So that's one covering each key event. But as I presented at the beginning of the talk, we know that there are other assays that measure the same key event that are also within the same test guideline. So these are being evaluated to see if these can be used within the same defined approaches. And I've just listed some of them here. So for key event 1, it's the other two that are in 442C, ADRA and EpiSense. For key event 2, it's Lusense. And for key event 3, it's Usense and Guard Skin. And there's also a couple of in silico tools also under evaluation for use in ITS. So iSafe Rat, Late Scope Model Applier, and StopTox. But looking now towards 3D skin models, so now moving away from some of the simpler assays into more complex ones, and I just wanted to highlight EpiSense A. So this is a test that was just published under test guideline 442D, so that covers key event 2. And this is a 3D reconstructed human epidermal tissue model. And this allows the direct application of test material to the tissue. This uses a gravimetric approach, which can mean that it increases the applicability domain or reduces the limitations associated with some of the other assays, and it's capable of predicting both hazard and potency. And I've just popped a reference here that shows its performance against some difficult to test chemicals, including lipophilic and hydrophilic chemicals, including pre and prohaptins. What we can see here is that this assay performs incredibly well for these. Now moving on to another 3D skin model. This here is a census, and this assay is actually under evaluation at the OECD. It also uses a 3D reconstructed human epidermal tissue model, and again allows direct application of the test test material to the tissue, and uses a gravimetric approach. This method also predicts hazard and potency. And I've just shown here in a couple of these boxes the performance when we look at potency for census. So this paper here shows 174 materials were assessed using the census, and the performance is here. So we have a balanced accuracy of 83%. So that's really, really good performance there for the census assay. And I just wanted to highlight its use for some more difficult to test chemicals, such as botanicals. So there's a paper here that I've cited that provides some information around how to use the census for botanicals, which traditionally have been quite difficult to assess. So again, the accuracy here, 96%. So seems like census is a really great option for some of these more difficult to test botanicals. And then moving on towards the end of the talk, since 2016, there's been a lot of movement towards developing approaches that provide something beyond hazard or potency in a categorical manner. What is really needed for a lot of risk assessment is a point of departure. And this table provides an overview of some newer defined approaches that are available for quantitative risk assessment or can be used to derive a point of departure. Now, this isn't an exhaustive list, but I've just highlighted some here. We've got a neural network published by Shiseido, and I've put some references here if you want to look further. We've got a 2 out of 3 regression model, the SARA model, which Emily will talk about in more detail next, and the Bayesian network model. And all of these provide a continuous output that you'd be able to use as a point of departure. But as I said, Emily will be talking more about SARA. So with that, I will stop and thank you all for listening before I hand back to the organisers. Thank you.
Thank you, Donna, for that great presentation. We're going to hold all questions to the end of the next presentation. We got lots of hands clapping. Great job. But please add any questions that you have right now to the Q&A module as they come up, and note if they're for a specific speaker. Our next speaker today is Dr. Emily Reinke. Dr. Reinke is a principal predictive toxicologist at Innovative RTP, where she provides scientific and operational support to the National Toxicology Program, Interagency Center for the Evaluation of Alternative Toxicological Methods, or NICETM. Her primary focus is on development and acceptance of alternatives for skin and respiratory sensitization and acute systemic toxicity. She received her doctorate in pathology from the University of Illinois, Chicago in 2013, and then worked for 9 years at the U.S. Army Public Health Center as a biologist, co-leading the In Vitro Screening and Toxicity Assessments program. She joined Innovative in 2022, and Emily has served on review panels with the Defense Threat Reduction Agency, US EPA, Japanese Center for the Validation of Alternative Methods, or JACVAM, and on independent peer review panels, as well as several OECD projects related to sensitization. She served as a Department of Defense representative to the Interagency Coordinating Committee on the Validation of Alternative Methods, or IIVM, for 7 years, which included 4 years as co-chair of the committee. Dr. Reinke is a diplomat of the American Board of Toxicology and is co-author of 16 peer-reviewed articles, 4 book chapters, and 27 technical reports. With that, please go ahead, Emily.
Thank you so much for that lovely introduction, Rachel, and thank you so much to the organizers for the invitation to speak today. As I said, I am a principal predictive toxicologist at INITIV, and I'm a contractor supporting the National Toxicology Program Interagency Center for the Evaluation of Alternative Test Methods. And standard disclaimer, these are my own views and not those of NICEDM or the NIIHS. Here we go. I can just briefly remind you of what the skin sensitization AOP is. Donna did a fantastic job of covering that and how these different key events are tested for with the different in vitro, in silico, and in vivo approaches. And what I will be doing is showing how we can actually take this forward into a risk assessment paradigm using computational tools that integrate all of these data together to make that prediction. So as Donna covered, there are several different kinds of defined approaches out there, and which she already talked about, the integrated testing strategy, the 2 out of 3 consensus. Rachel briefly touched on the sequential testing strategy, all of which can be used with the in vitro methods and potentially an in silico method to help integrate that. But these are predicting either hazard or a potency estimate for GHS subcategorization. Where some of these other models that are listed in this bucket come in, such as artificial neural networks, Bayesian networks, support vector machine, and meta models, is they're taking more of a computational approach to define that continuous curve to predict a point of departure. Here's a slightly more detailed table from what Donna presented earlier. Again, these are kind of showing the comparisons between those DAs that can predict potency, such as the ITS V1, V2, and the STS, which again are bringing forward that GHS category 1A or category 1B subcategorizations. You can see where those have been applied within regulatory use. The other models that are out there, such as the Bayesian ITS3, the Shiseido ANN, the 203 regression, and SARA-ICE, all are taking in various different in vitro or in vivo methodologies, as well as physical chemical properties for some of them, and deriving either an LLNA-like outcome, so similar to an EC3, or in the case of SARA-ICE, it is a human relevant prediction for that point of departure. The other difference on these lower 4 is that they actually are, again, providing that dose per unit area. And so that's where you're able to apply this within a risk assessment, is you can take that point of departure and then take that forward into your risk assessment in order to determine if even a skin sensitizer within a final product is going to be an issue because its concentration is low enough that it will not be a concern. So 2 of the models that I wanted to cover today briefly, because they are interrelated and we've done evaluations with both of them with a similar chemical set. First is the artificial neural network. And so neural networks behave similar to how we think of how our brain behaves, and that you have multiple nodes that are interconnected in order to form this network that can then make an outcome. So for the artificial neural network from Shiseido, you have multiple input layers such as the log P, the key event 1 test, so DPRA in this case, the ARE assay, keratinosins, and HCLAT. Multiple iterations are run with this neural network to try and make a prediction, and then you have an LLNA threshold percentage, which is that EC3-like outcome. These can be translated into those different potency classes, or again into a GHS subcategorization, and also used for a risk assessment. Hirota et al. Produced this model initially using proprietary software QuickNet as part of an IT case study with the EPA. This was reproduced in our studio, and you can see here on the bottom that, let me get my laser pointer. There we go. So you can see here how well the Hirota and the R models recapitulate each other, so showing that they are both coming out with the same outcome. There are two separate models for the ANN, one that only uses DPRA and HCLAT as its in vitro inputs, and then another that uses DPRA, HCLAT, and keratinosins as its inputs. The other model that I want to speak about today is the Sarah Ice model, which I will actually be spending a little bit more time on because this is under evaluation at OECD for inclusion in test guideline 497 for that derivation of a point of departure. And so it's going to be the first example of a model that can derive those points of departure for use in risk assessment at OECD. So Sarah Ice is a Bayesian model. And so when you think about what a Bayesian model does, it's specifying a possible set of outcomes based on an a priori judgment. And so what you have is you have all these different input types. You have your LLNA data that you can put in, not that we're recommending that you do any more LLNA testing, but if you have historical data that you could use for this, if you've created DPRA, HCLAT, keratinosins, USENS, any of those can be input into the model. They're going to interrelate against each other. You're going to integrate your human data that you have. Using Bayes' theorem, you can then come out with a probabilistic output of where you think that EDO1 point of departure is going to lay. And so here you can see that you get this nice Gaussian curve that then shows where we think that this outcome is going to be based on the weight of evidence from that input of your different types of data. The output of Sarah Ice is termed the EDO1, which is a 1%, the dose at which a chemical is expected to sensitize 1% of a human predictive patch test eligible population. So this would be, if you were to apply this in an HPPT test, 1% of that population would be expected to be sensitized at that dose level. A little bit of history of where Sarah Ice came from. The skin allergy risk assessment defined approach was initially developed at Unilever to be used within a weight of evidence next generation risk assessment framework. And so the idea behind this is that you can take all available data, whether that's in vitro data, in vivo data, physicochemical properties, use cases, exposure, any of that can then be fed in to the model, and you get an output of that, again, that EDO1 with an exposure estimate in the case of the Sarah DA. So you have, again, you have your different, you have your different inputs that you take there. You then have that Bayesian model applied, and you can do your risk assessment conclusion from there. And again, it's still that EDO1 that is the output. Sarah Ice has been developed since about 2017, and with initial publication in 2019 by Joe Reynolds. Updates were done in the 2019 to 2021 period. In 2021-2022, a new model was put out, and at that point, this is when Unilever and Nicedum partnered in order to further develop Sarah into a more applicable model for generalized regulatory use. So one thing that to bear in mind with the Unilever model is that it was for consumer use products, primarily for cosmetics. And so they had a lot of additional information that they were going to be able to use for their own specific internal uses that would not be applicable outside of Unilever or very other specific use cases. So this was in order to make the model more applicable in a general sense. So what was done to modify Sarah into Sarah Ice? And that ICE stands for the Integrated Chemical Environment. And so one of the primary tools that we have out of Nicedum is the Integrated Chemical Environment. It's a fantastic resource for providing toxicity data, user-friendly ways to do IIVE, PBPK assessments, chemical searching to try and find similar chemicals. If you want to do that read across approach, all kinds of different things in that tool. So if you're not familiar with ICE, the QR code is here, and it is definitely a tool worthwhile to look at if you have not looked at it yet. So what we did is we took the ICE database that all of our skin sensitization data, all of for the in vitro models, LNA models, as well as human predictive patch tests that we had from a project that was ongoing with the TG497 development. And we added this into the Unilever sera model. The other thing that was removed was we de-emphasized the risk benchmarking component of the model. And this was done because for the expanded chemical domain within the sera ICE database, we didn't have those use estimates and exposure assessments for this wide range of chemicals. And so it would be really hard for the model to automatically predict that, not having those inputs to be able to train it. So that was removed from the model. And then additionally, the ability to apply a GHS classification was added into the model as well. And so you can take that EDO1 and the fact that this is a probabilistic assessment and determine whether or not a chemical will be a 1A, a 1B, or an NC based on that weight area under the curve of that output. So how does the sera ICE model work? Well, you have your different inputs. You can do, again, HPPT data, and this is our catch-all term for human repeat insult patch tests, as well as human maximization tests. So if you have that human data, you can put that in. You can also put LNA data in there. Again, not recommending that any additional LNA data are produced, but if there's historical, then you can add that in. You can also provide in any of the in vitro assays that the model has been trained on. So that includes the DPRA, the kinetic DPRA, keratinescens, USENS, and HCLAT. The sera ICE database is predicated on 434 chemicals. So it's actually a really robust database for training this model. You take your input data, it is then applied to the statistical model, which again is that network of probability distributions to describe the associations between all the data types. So it's going to figure out that based on the a priori predictions, what it's going to, what that distribution is going to look like. You then can get that EDO1, again, that's that 1% sensitizing dose in an HPPT. And then we can apply the GHS classification thresholds from 1A, 1B, and 1B NC onto that. And we can make a determination based on, again, where that weighting is of what subcategory it's going to be. The model assumptions that are involved in sera ICE, what is that there's a dermal dose at which there's a 1% chance of inducing sensitization in a randomly selected population. The probability of inducing sensitization increases with dose. Each individual has their own threshold. The distribution of personal thresholds has a Gaussian shape, so normally distributed. And that different chemicals have different variabilities within that human population with respect to the sensitivity for induction to sensitization. And that the number of individuals sensitized in an HPPT study follows a logic normal binomial compound distribution. The non-HPPT data, so that's going to be the LNA and any of the other in vitro inputs. These assumptions are that these data can be transformed such that you can have a normal distribution. So how is that going to look? The same transformations when put on a scale in which it's reasonable to assume linear relationships is similar to that of for the log EDO1. The relationships between the average results can be described by a multivariate Gaussian distribution. Variability in each test is chemical specific. So the test itself doesn't have a wide variability, but the chemical might have some. There is a latent variable for each test and each chemical, which defines the variance of the chemical in that particular test. And then chemical specific variance parameters can be estimated using partial pooling. This means that the population of variances for each tested can be learned and used to regularize chemical specific estimates when limited data are available. So a little bit of the breakdown of what sera ICE looks like, again, from the databases. This is what was used to train the model. Again, we have 434 distinct Cas-RN within the model itself, encompassing well over 3,000 studies. So, and you can see from this table how they're distributed across the different input parameters that we can use. So you can see with the HPPT, 276 unique Cas-RN with 871 studies. One of the key features that you have to have in order to build a Bayesian model is that you have repeat data so that you can, again, have that understanding of variability. within the methods. And so with all of these, we do have really good repeat data for all of the chemicals that were put in. Again, Sarah Ice, it's a computational model, which means theoretically you could do this on pen and paper. I don't think anybody would like to do it on pen and paper, but you can. The model was programmed in Stan, and then Python is the language that we've used to actually process model inputs and outputs. However, again, not everybody is able to use Python. I certainly cannot use it. I look at coding and kind of panic a little bit. And additionally, in order to run it in Python, it actually requires many CPU cycles in order to get this output. So what we've actually done is that we have a production model that can take this model and apply it in a more friendly manner. And what that looks like is we actually have this Sarah Ice tool. This is downloadable as an executable file right now. We are working on moving it into a web application that will be, won't even require the installation, but can still be run independently of the internet. So it can be on a firewall computer if that's what's needed. And we are working on developing this and putting it out so that anybody can use Sarah Ice very, very easily. And what we can see from what this model looks like, you have your chemicals of interest, you have your input data, which for these chemicals, I can't remember what we had for input data, but you get a mean EDO1 as your output. You get your different percentiles, because remember, this is a distribution. You also get your probability of GHS subcategorization. And then you can see how this distribution for the chemical looks across the, low probability versus high probability. So what we're looking at here is we have the 90th credible interval, which is the thin gray line. You have and that's from the 5th to the 95th percentile. You have the thicker gray line, which is from the 25th to the 75th percentile. And then you have the median is actually what we have modeled here. And then you can also see the vertical yellow line is the 1A, 1B cutoff, and the vertical purple line is the 1B NC cutoff. So you can see all of these fall very strongly within the sensitizer realm. We could say this top one, just glancing at it, you can tell it's a 1A, and that it's probably going to be a fairly extreme sensitizer just by looking at the distribution. And then you can come over here and you can look at the mean EDO1 and actually get an idea of what those dosages look like for that sensitization level. Again, we apply the GHS classification onto the model as well. And so we're using that threshold of 60,000 micrograms per centimeter squared as our total cutoff, which means that, anything above that is going to be is an NC by GHS categorization. And this is the highest possible dose under a standard HPPT assay. And then we also use that threshold of 500 micrograms per centimeter squared to define the boundary between 1A and 1B. And then we use the area under the curve between those thresholds to determine the probability mass to that attributable to that interval. And so as you can see here, the probability mass on this chemical is very, very heavy within the 1B to NC range. And so it's most likely that it's going to be a 1B chemical. How does Sarah Ice perform against different benchmarks? So what we've taken is we've compared Sarah Ice to the TG497 classic chemical list, which there's 197 chemicals in there. And for inputs, we only used a single in vitro instance for DPRA, keratinosense, HCLAT, and KDPRA. So you can put multiple tests in if you have multiple tests. And you can see that we have a balanced accuracy for binary within Sarah Ice of 94% against the human data. And then we have a balanced accuracy of 91% against the LLNA data. So the model performs really well for hazard classification. And then when we look at the subcategory classifications, again, this is using those single in vitro inputs. We can see we have an average balanced accuracy of 82% against the human data, and an average balanced accuracy of 83% against the mouse data. Again, these are averages. You can see how well it performs for the different subcategories. It struggles a little bit with the 1Bs, but some of that is also related to how stringent we have made our cutoffs for that subcategorization. And one of the things that's available in the downloadable file to run SARA-ICE is you can actually adjust your probability thresholds in order to say, you know, I really want a stringent threshold versus a not so stringent threshold. So now to the actual case study, now that I've actually explained SARA-ICE to you, and I'm sure I probably confused the subject a little bit too, because also not a computational modeler. The isothiazolinones are biocides that are used in material preservatives to prevent the growth of microbial organisms. And the reason we're interested in ITs is EPA came to us several years ago asking for help in trying to do a risk assessment for these chemicals specifically. And what we actually used at the time was the artificial neural network. And for those IT compounds, we collected and analyzed all available in vivo data, and then using the in vitro methods that at the time were the only ones available within the guidelines, so the DPRA, the H-clat, and the keratinosens, produced in vitro data for each of these compounds. And then also used the OPERA suite to predict the physicochemical properties for each of those. And using the artificial neural network, we are able to produce these different outputs that are similar again to that EC3. And this is important because we can now use everything in this column as a comparator against how the SARA-ICE model is performing for each of these different ITs as well. So we can use these same inputs, add additional data that have been made over the subsequent years, and actually make these predictions to see how they compare to each other. So I realize this is a very hard to read slide. We'll zoom in a little bit to a specific chemical for interest. But what we've done here as part of the paper that was just accepted, I see that says under review, that should actually say accepted now. We have actually looked at these different ITs using different data inputs within SARA-ICE to see how, again, the width of the distribution changes and how the sensitivity of the assay output changes. And so we can see with all of these different assay inputs that the chemical becomes either more likely to be a sensitizer or less likely to be a sensitizer, and the distribution changes with the amount of data that's available in there. And just as a note, we have several reference lines on here to allow the reader to understand how these compare to other points of departure that have been used for risk assessment. And so we have the red line is the NESSL, that no expected skin induction level. And so that's a level at which you would expect no skin sensitization to occur at all in any part of the population. A green line is the medium-like location parameter for the LLNA. So that's a summary statistic for the LLNA for all available data. And again, that's that EC3. And then the blue vertical line is actually the point of departure that was derived from the artificial neural network case study by EPA. And so you can see we've got those comparisons on here to see how SARA-ICE is performing. To zoom in a little bit more closely, we're going to look at MIT. And so here are the types of input data that we were able to actually put into SARA-ICE to do this assessment. We have multiple DPRAs, 1K DPRA, multiple keratinosens, H-clat, USENS, and LLNA data. And so we're able to put that into the model. And again, I'm going to go back to that same figure, but we're going to zoom in a little bit. And we can see how with those different data types, how the predictions, again, that spread either widens or narrows, and how that median moves to the left or right. So left is going to be more potent, right is going to be less potent. So we have that if you have only in vitro data, you're going to have the most conservative estimate, but you have a bit of a wider spread because we only have, a few data points. If you do, just the LLNA or the HBPT, you can see that it goes to the left, and both the medians on those are actually to the right of the EPA POD and the LLNA EC3. And so, but it's also nice to see that it's predicting fairly close to what those predictions are. And then as you add more data in, you can see that the distribution narrows quite a bit. So again, just the same thing to look at that as a more highlighted, where you can see the different types of how the different types of outputs change the EDO1 prediction. So the other thing that we can look at is we can see the different percentiles that are output by that. This no data is literally you're going to get the exact same output for the model regardless of what chemical you're interested in, because it's chemical agnostic. It is input, requires the inputs. But you can see like here you've got that mean EDO1 of 9.8. It's way more conservative than something like where you've only got the LNA at 440. Regardless, we can tell that based on this, that MIT is going to be a GHS category 1A based on that probability distribution of the of that output as well. And so you can see, it's for almost all of these, you're going to see that it is highly likely that it's a 1A. One thing to note, though, is that the model has a hard time with it does not actually predict a subcategorization for the LNA inputs only. It's actually not sure if it's a 1A or 1B. It knows it's a sensitizer, but it can't make that prediction. So this is just highlighting that in vitro data is really useful in helping to have a better prediction out of the model. One thing that you can do that Sarah Ice, the model, it cannot do alone, but that you could actually apply is you can actually take those probability distributions and you can predict at which exposure levels, so here's an exposure micrograms per centimeter squared, are you likely to be safe or unsafe for those different EDO1 distributions. And so what we see here, anything in blue is a high probability that you are not going to have an exposure greater than the EDO1. Everything over here in orange is likelihood that you are going to have an exposure greater than the EDO1. And then this gray area is where the model is uncertain. So you would expect some amount of uncertainty kind of in that area where you're really, really close to the EDO1. And so this is how it could be applied within a risk assessment is you can do something like this. You could take the geometric mean of the EDO1 and use that and apply different safety factors if you need to do that. Again, this is a human relevant output, so you don't necessarily need to think about too many different safety factors as opposed to like an LLNA output, but you would probably still need to have a few because it's not an actual human exposure. So you can, again, take that however you want to, part of the distribution that you want to pull out is what you can pull out. So with that, I'll wrap everything up on this. And like I said, I'm sure I probably confused a couple of things, but we can clear it up in the Q&A. Sarah Ice is being adapted for regulatory use through expanded data and functionality, and it will be the first probabilistic defined approach included in any OECD test guideline. The DA shows really good concordance with sensitizer binary and GHS subcategory classifications against OECD benchmark data. We've done several case studies. So in this case, we've done the MIT case study. We also have some fragrances case studies ongoing, as well as a few others with EPA and other federal agencies to look at how Sarah Ice can help them in their risk assessments. And with that, I will acknowledge the Unilever team and my NICEDM team, and we can open the floor up for questions. Thank you.
Thank you, Emily, and thank you, Donna, for two great presentations. We have quite a few questions in the Q&A, so we'll try to get through as many as we can. I wanted to mention that Donna has been very busy responding to some of those questions already, and we've actually got people attending responding to questions as well, which is very cool. So if you want to see some answers to some of these questions, go ahead to the Answered tab and you'll be able to see those written answers. Okay, so the first question we have is for you, Donna. It's for the ITS version 1 or 2, there's an option to come with a conclusion if the in silico result is out of domain. For example, if a prediction can't be made, if the chemical is non-discrete, can the classification still be accepted based on the DPR and the H-CLAT?
Yeah, so thank you for this question. I think it's really helpful to clarify these with the ITS because it can be a little bit more complicated. So I'll try and split it into two parts. So you mentioned at the end, assuming that they are non-concordant, but just as a reminder, the ITS uses a scoring system, so there's no concordance or discordance with the results from the assays. The scores are just used to assign the potency classification. And with respect to using the partial information sources, in terms of whether it can still be accepted, if you mean accepted by a regulatory body, then that's not something that I can answer. But the fact that the use of partial information sources is published in Guideline 497, so where we have an out-of-domain in silico prediction, so your classification is based only on HCLAT and DPRA, in my opinion, there should be no reason for a regulator to say, no, they can't accept it because it is in the guideline as an accepted way to use the ITSDA.
Okay, great. Thanks, Donna. So the next question is, when it's for Emily, when will Sarah Ice be available for broader evaluation / validation by stakeholders to evaluate particular classes of chemicals, for example, fragrances?
I wonder who I got this question from. So we have actually already done some evaluations with fragrances and Sarah Ice. I could probably try to find the slides really quickly, but we have been partnering with RIFM in some cases and looking and trying to pull all of their nestles that they have and any in vitro data that they have and run those in the model to try and have that comparison. Again, comparing the nestle to the EDO1 distribution or the geometric mean or whatever output we want to talk about from that isn't 100% appropriate, just given that one is for a no induction level at all, whereas the other one is actually supposed to be that 1% sensitizing dose. But we can use that as kind of a benchmark for how it's performing. So we have done that to some extent. We haven't finalized it or anything along those lines, but in terms of that, it is ongoing. For the model itself to be available, you are more than welcome to send me an email and I can go ahead and give you access to where the model is currently stored for beta testing. But I would highly recommend waiting until we have the latest update done. It has already undergone some evaluation through ICCS partners, as well as through the OECD Defined Approaches Risk and Sensitization Expert Group. So we've already done quite a bit of evaluation with outside partners to make sure that it is performing to their expectations. And it has been validated already against a very large chemical set to some extent as well. So we're always looking at new ways that we can look at how the model is performing. So with that being said, you can send me an email and I can put you on our list and let you know when the latest version gets released. But the version that is currently available and has undergone the initial evaluation is getting updated. And so I wouldn't want to, I mean, it works just fine, but the user interface will be a little bit better in the next version.
Thanks, Emily.
Great, and maybe just sticking with Emily and the Sarah Ice model. There are a couple questions, Emily, just focusing in on the applicability domain. So for example, only single chemicals or also formulations. There was a question that Donna answered related to pesticides, but maybe you could elaborate on use of pesticides and pesticide formulations as well.
Yeah. So technically it has not been evaluated against formulations. So I would say the same caveat that goes against all the other OECD test guidelines of you can use it with caution. If you have formulation data, potentially something could get predicted, but again, it has not been formally evaluated against formulations, and the database that it is built in is on single ingredients. It does contain pesticides in there, and so for that applicability domain should apply, as well as for some again, it's industrial chemicals, cosmetics, other personal care products, etc. The full database is going to be available online within the next day or so on the NICEDM website. We've been doing a lot of updating to our websites as we're going along, as well as it will be included in the OECD test guideline when that is eventually approved, and the paper that was just accepted recently will also have the database in it. So you can go in and see exactly what chemicals are in there. I can't remember all 434 off the top of my head.
Christ, you can't. All right. So this question could probably be for either of you, but maybe Donna, at least to start. So when using the updated DAs, which allow the use of multiple NAMs, do you expect there will be any guidance in future versions of OECD 497 as to which assays are most applicable for a particular test chemical, or is this up to the user?
Yes, thank you for the question. So if in the future the alternate methods are accepted and are added to guideline 497, I think right now I don't foresee there being guidance in the actual guideline itself, but this may of course be published elsewhere. And with respect to the assay choice, I think as is always the case, the limitations for each individual assay as detailed in 442 C, D, and E for the Ds that I was talking about, then that should always be considered before you use them within a defined approach. I don't know if you've got anything to add, Emily?
No, I think you pretty much said it.
All right. There's a question for both of you that I asked for clarification on, and I've got that clarification, which is that this person is wondering about the data that's been used to build some of the computational models that you talked about. And they want to know if that data has been chosen according to any standards to ensure quality. I guess that would also apply to the chemicals used to evaluate the defined approaches that you presented as well, Donna. But maybe each of you can speak a little bit about what kinds of data quality procedures go into building some of the models.
So Donna, I can hop in. One of the things that NICEDM particularly prides itself on, and that the contract supporting it makes sure we do, is all data that go into the integrated chemical environment have undergone a very robust and rigorous QA evaluation prior to going in. And anytime anything is caught that maybe we missed, we update it as soon as we possibly can in the backend database. So all of the data that went into the Sarah ICE model database did undergo a very thorough QC. We cross-check it against the original source. We make sure that we have multiple people checking to make sure that the numbers are appropriate and correct with what is against the original source. If we have multiple sources citing the same number, we remove those duplicates so that we don't have that duplication that could cause issues with the model development. And then, we make sure that, again, that everything is as clean as possible before it goes in. And so that same robustness was used for the Sarah ICE database. I can also say for the human predictive patch test database that was used, an even more thorough evaluation went in terms of the study quality prior to being even included in the HPPT database. And there was a 5-point system that was used about whether or not a study even met standards for a guideline application. So I would say that we've been as thorough as we could.
Thanks, Emily.
And yeah, just to add to that, if you're referring to the in silico models used in the ITS, then similar to what Emily's just laid out, the approach to standardization of the data that's input into the in silico models is extremely robust. But what I will say is that I read this question a little bit of a different way, and I wasn't sure if it was referring to the inputs into the defined approaches. So I also want to answer if that's the question.
Yeah, go ahead.
So if the question is, how do you standardize or how do you assure quality of the inputs that go into the DAs, then for the 2 out of 3 and for the ITS, then those inputs are OECD test guideline methods. So that quality assurance is already there. It's been run to an OECD test guideline. So there should be no reason to doubt the quality. It'll have been done in a GLP lab, etc. And then if you're referring to the quality of the in silico, then of course right now for ITS, it's only DEREC and OECD Toolbox that are permitted to be used in the ITS. And the information around the quality of data, its training set, how it performs in a test set, is all contained or cited in Guideline 497 with respect to the protocol, the QSAR model reporting formats, the QMRF, and of course they both fulfill the OECD's QSAR 5 validation principles. So all that information is in there. So yeah, I answered that in a few different ways. Wasn't sure exactly what the question was referred to. So hopefully that's answered to the question.
Yeah,
and I was going to add on a little bit to that too. Reminder that just because something was done in an OECD test guideline and it qualifies under MAD, it doesn't mean that you have to accept that a regulator has to accept the data either. So they are going to be looking at the individual inputs into the DA, and they're going to evaluate whether or not it met guideline requirements and is an acceptable study. So yeah, as Donna said, everything, if it's done as a test guideline, it's going to be done under GLP. It's going to be done with all those documentation requirements for that. But that doesn't necessarily mean that a submitter putting it in, and Rachel can correct me if I'm wrong, but a submitter saying, in a 203, this is a hazard, they can look at it and go, well, I don't believe that this DPRA was a legitimate assay. So you're missing a data point.
Thanks, Emily.
Yeah, thank you both.
I was just going to say, yeah, that's
okay.
absolutely correct. Emily, like we look at the input data and then we make our own interpretation. So yeah.
Thanks, Rachel. Let's see. So I see Christy's typing an answer to one question, and then there's just one remaining that we can get to in the last couple minutes here. And this is for you, Emily. Can you please explain again the accuracy of the Sarah Ice method? And the question continues to ask if the results were only compared to single in vitro methods.
Okay. So in terms of the accuracy of what I showed was against the either binary or GHS subcategorization were the two tables that I showed. And those were compared against human data. So again, was within the test guideline 497 database, we know whether a chemical is a sensitizer by the human test, and we know whether the chemical is a sensitizer by the LLNA. And so those were the comparisons that we made. So either against the human binary or the human subcategorization, or the LNA binary or LNA subcategorization. In terms of performance, when you think about the entire distribution, that's a little bit harder to do. We do have that data in the paper coming out. I didn't show it here, but in terms of like a regression for it, it is fairly accurate. I can't remember off the top of my head what that number is, but it does perform really, really well when you think about it across the entire distribution when trying to compare it to other similar outputs. Again, it's really hard when you have a full distribution to compare something to a single point.
Great point. Thanks, Emily. Well, we have gotten through all of our questions, which is great. So I want to thank the speakers for sharing your work with us. Thanks to all of my co-hosts. I want to point out something new for this webinar. There's a survey that should pop up in your browser when the webinar ends, and we'd really appreciate getting some feedback from you. So if you would take a moment to fill out that short survey, that would be great. And I will go ahead and close the webinar and wish you all a wonderful holiday season. Thank you.
Thank you.
Thank you.
Thank you.
Bye, everyone.